Muscle spindles are sensory receptors within the belly of a muscle that primarily detect changes in the length of this muscle. They convey length information to the central nervous system via sensory neurons. This information can be processed by the brain to determine the position of body parts. The responses of muscle spindles to changes in length also play an important role in regulating the contraction of muscles, by activating motor neurons via the stretch reflex to resist muscle stretch. Muscle spindles are found within the belly of muscles, embedded in extrafusal muscle fibers. Note that fusus is the Latin word for spindle. Muscle spindles are composed of 3 to 12 intrafusal muscle fibers, of which there are 3 types: dynamic nuclear bag fibers, static nuclear bag fibers, nuclear chain fibers, and the axons of sensory neurons. Axons of gamma motor neurons also terminate in muscle spindles. They make synapses at either or both of the ends of the intrafusal muscle fibers and regulate the sensitivity of the sensory afferents, which are located in the non-contractile central region. Muscle spindles are encapsulated by connective tissue, and are aligned parallel to extrafusal muscle fibers, unlike Golgi tendon organs, which are oriented in series. The muscle spindle has both sensory and motor components. Primary and secondary sensory nerve fibers spiral around and terminate on the central portions of the intrafusal muscle fibers, providing the sensory component of the structure via stretch-sensitive ion channels of the axons. In mammals including humans, the motor component is provided by up to a dozen gamma motor neurons and to a lesser extent by 1 or 2 beta motor neurons. Gamma and beta motor neurons are called fusomotor neurons, because they activate the intrafusal muscle fibers. Gamma motor neurons innervate only intrafusal muscle fibers, whereas beta motor neurons innervate both extrafusal and intrafusal muscle fibers and so are referred to as skeletal fusomotor neurons. Fusomotor drive causes a contraction and stiffening of the end portions of the intrafusal muscle fibers. Fusomotor neurons are classified as static or dynamic according to the type of intrafusal muscle fibers they innervate and their physiological effects on the responses of the ear and 2 sensory neurons innervating the central, non-contractile part of the muscle spindle. The static axons innervate the chain or bag 2 fibers. They increase the firing rate of ear and 2 afferents at a given muscle length. The dynamic axons innervate the bag 1 intrafusal muscle fibers. They increase the stretch sensitivity of the ear afferents by stiffening the bag 1 intrafusal fibers. Sensitivity modification. The function of the gamma motor neurons is not to supplement the force of muscle contraction provided by the extrafusal fibers, but to modify the sensitivity of the muscle spindle sensory afferents to stretch. Upon release of acetylcholine by the active gamma motor neuron, the end portions of the intrafusal muscle fibers contract, thus elongating the non-contractile central portions. This opens stretch-sensitive ion channels of the sensory endings, leading to an influx of sodium ions. This raises the resting potential of the endings, thereby increasing the probability of action potential firing, thus increasing the stretch sensitivity of the muscle spindle afferents. For an interactive animation created by Jan Kowalczyk at the University of Alberta, demonstrating spindle afferent responses to muscle stretch with and without gamma action, go to Arthur Prochaska's lab, University of Alberta. How does the central nervous system control gamma fusomotor neurons? It has been difficult to record from gamma motor neurons during normal movement because they have very small axons. Several theories have been proposed, based on recordings from spindle afferents. 1. Alpha-gamma coactivation. Here it is posited that gamma motor neurons are activated in parallel with alpha motor neurons to maintain the firing of spindle afferents when the extrafusal muscles shorten. 2. Fusomotor set. Gamma motor neurons are activated according to the novelty or difficulty of a task. Whereas static gamma motor neurons are continuously active during routine movements such as locomotion, dynamic gamma motor neurons tend to be activated more during difficult tasks, increasing ear stretch sensitivity. 3. Fusomotor template of intended movement. Static gamma activity is a temporal template of the expected shortening and lengthening of the receptor-bearing muscle. Dynamic gamma activity turns on and off abruptly, sensitizing spindle afferents to the onset of muscle lengthening and departures from the intended movement trajectory. Stretch reflex. When a muscle is stretched, primary sensory fibers of the muscle spindle respond to both changes in muscle length and velocity and transmit this activity to the spinal cord in the form of changes in the rate of action potentials. Likewise, secondary sensory fibers respond to muscle length changes and transmit this signal to the spinal cord. The ear afferent signals are transmitted monosynaptically to many alpha motor neurons of the receptor-bearing muscle. The reflexly evoked activity in the alpha motor neurons is then transmitted via their efferent axons to the extrafusal fibers of the muscle, which generate force and thereby resist the stretch. The ear afferent signal is also transmitted polysynaptically through interneurons, which inhibit alpha motor neurons of antagonist muscles, causing them to relax. After stroke or spinal cord injury in humans, spastic hypotonus often develops, whereby the stretch reflex in flexor muscles of the arms and extensor muscles of the legs is overly sensitive. This results in abnormal postures, stiffness and contractures. Hypotonus may be the result of oversensitivity of alpha motor neurons and interneurons to the ear and two afferent signals. PNF stretching, or proprioceptive neuromuscular facilitation, is a method of flexibility training that can reduce hypotonus, allowing muscles to relax and lengthen. Development. It is also believed that muscle spindles play a critical role in sensorimotor development. Additional images. See also, Type ear sensory fiber, Type 2 sensory fiber, alpha motor neuron, gamma motor neuron, beta motor neuron, intrafusal muscle fiber, extrafusal muscle fiber. References. External links, Muscle spindles at the U.S. National Library of Medicine. Medical subject headings.